do as well, really. And it gives aesthetic to your device as well. It makes it look more organized and more stable. And on top of that, you have an automated pneumatic valve controller. You have your electronics for your operating physical sensors. Your potentiostat is also there for measuring, of course, your electrochemical signals. And then a computer for central programming integration of all these commands. The lower image shows an schematic of the integrated microfluidic device consisting of your modular components, including your microbioreactors if you wanted to incorporate that, your breadboard, your reservoir, your bubble trap. So bubbles are quite a common, stressful operation in microfluidics, and you have like trap bubbles. And then you have physical sensors and electrochemical sensors as well that you can incorporate. The small inset there shows basically a photograph of the actual integrated platform that you see in the schematics. So those two are the most recent and most useful and advanced methods that you can incorporate to make your MOCs capable of long-term monitoring and sensing. Now let's move into discussing actually some of the recent studies and recent microfluidic devices that have been developed for drug discovery and drug screening. So next slide, please. Thank you. So as you are aware, drug discovery and screening have been the biggest hype. So the last few months with this pandemic that put Earth on a halt, especially the Philippines. However, despite the successes and critical advancements in developing various approaches over the past decades or maybe past 3 decades, it is increasingly recognized that the preclinical stages of our current drug development pipeline have failed to fulfill the requirements of accurate predictions of drug responses and their extrapolation to humans. So one of the reasons probably here is because the pathological mechanism that has been observed between animals and humans are quite different. So I think since the generation of my Lola, several cell culture systems in vitro were widely used, so they don't have the microfluidic system yet then. Although these techniques, these cell culture techniques have allowed for drug discovery studies and disease modeling, conventional 2D culture systems, as I mentioned before, in which the cells can be cultivated in a monolayer, fail to replicate the biochemical environment in vivo and other mechanical properties as well. So this is very important, and often biologists sort of not really ignore, but forget about the mechanical properties as well inside your biological system. Moreover, so drug diffusion kinetics cannot be demonstrated accurately in 2D cell cultures, right? And so it's really hard to demonstrate diffusion kinetics in a 2D system, where the drug doses are effective in 2D, but universally manifest as being ineffective in real human body. And so it makes complete sense. These culture models usually do not maintain their differentiated cell functions as well. So those are disadvantages of cell cultures. If you think other disadvantages, feel free to ping me. Those are things that I could remember when I was discussing about insisting on using microfluidics than the typical cell culture for a drug screening study. Anyway, to address the lack of physiological relevance, which is the major drawback of these 2D cell cultures and even 3D cell culture models. So from 2D, you have 2D cultures that have gained attention with the improved tissue organization and enhanced expression of cell function. But even that is not enough. Even optimal 3D culture models also suffer from a shortcoming of reproducing the characteristics of living organs, which are crucial for their functions, including tissue-tissue interfaces, temporal and spatial gradients of chemicals and oxygen, and again, the mechanically active microenvironment. Hence, we have preliminary investigations in vivo using animal models. So we move from 2D, 3D, and then we look into animal models as the gold standard, and remained absolutely a necessary step, at least in US FDA, and I believe in the Philippines as well, the FDA, necessary step in drug development process. However, all these three have a lot of limitations. This is primarily due to their ability to maintain the significant intricacies lying in living systems, an ability to do that. So, and also an ability to evaluate organ-to-organ crosstalk and allow for the determination of pharmacological attributes as well as toxicological issues. However, these models, like I mentioned before, have big limitations in terms of phylogenetic discrepancy between laboratory animals and humans. We've seen this again and again in many research, which makes it difficult to observe and precisely extrapolate the effects and responses on inherently complex interconnected tissues, that in particular. Therefore, it is increasingly being recognized that preclinical assessments that are based on animal models often end with poor predictions in many cases, and we need definitely better models. So let's take a look at some of these recent body-on-chips which address these issues. So here in this study, I love animals. So using body-on-chips to emulate a disease or pathology is far less grueling than actually triggering a disorder like this one out of an innocent rat. Here, they look at ALS. So you see here the loss of, with ALS, the loss of neuromuscular junction, or your NMJ, is basically an early and critical hallmark in all forms of ALS. In this study, a functional NMJ disease model is developed by integrating motor neurons, or usually they abbreviate it as MNs, differentiated from your multiple ALS patients. Induced pluripotent stem cells, or IPs. I've discussed IPs, I think, a lecture before. And primary human muscle. So they also use primary human muscle into a chambered system. You can see it here. So the NMJ functionality is tested by recording the myotube contractions while stimulating your MNs, your motor neurons, by field electrodes. And a set of clinically relevant parameters is defined to characterize your NMJ function. So you develop a chip and then try to test it. In this particular illustration, you can see the NMJ system at the top. So that's schematic over there. Sorry, keep on pointing, but I know that you cannot see it because my pen is not working. The IPCS-MNs, your motor neurons, were co-cultured separately with your wild-type skeletal muscle, or your WT-SKM, where the MN chamber and SKM chamber were connected through microtunnels that were axonal permissible, but chemical and electrical impermeable. So that's very important. So your axons can actually travel through, but the chemical and electrical permeability is not present. So in figure A, you see clearly the cell plating scheme for the NMJ cultures inside your microchamber. So there's an illustration there. And then the diagram, so the image B, you have here the diagram of NMJ chamber system illustrating your motor neuron chamber and then your SKM chamber, which are connected by your microchannels. And through which, in this microchannels, through which your MNs sent axons to reach the muscle. And so it's illustrated quite clearly in red. You can see that. Your motor neurons were stimulated by field electrodes and induced by myofiber contractions, were also captured in this study by pixel differentials through a phase contrast microscope connected to a video camera. So I think I've stressed quite a lot about integrating your microfluidics with super resolution microscope, or even just a confocal microscope. So the advantage of microfluidic devices is that you can move it around. So you could couple it with different optical device and even non-optical device for measuring. Moving on, so you see at the bottom, so I think that's a face images. This is a face image of cells in your NMJ system, demonstrating that your motor neurons in the motor neuron chamber and myofibers in the muscle chamber. So the distinctions between your motor neurons and your myofibers are muscle chamber. So you have there, you see the red arrows that indicated the tunnel openings for your axons or axonal exits, and your yellow arrows, those point to some axons already in your muscle chamber. And then your image D is actually just an immunocytochemistry analysis of your NMJs in the chambers. So you have here an image from the muscle chamber indicating an axonal terminal. Red stained, so that's red stained with neurofilament branch at the terminal and wrapped around your microtube. I don't know if you can see that clearly, which you can visualize with the marker myosin heavy chain. So that's your green. So that you can see quite clearly. Hopefully you see the axonal terminal as well, the red. Actually, I think if my internet will allow me, I have a background here which actually shows you my microfluidic device and how do I do this? And then you see the neurons, but hopefully it doesn't fail me. So you have there the background. So let me just start the video. Hopefully it works and you don't see this limited space. So that's the neurons actually, that's the axon. So you see it firing really nicely. And you see, if you see this red lines, really bright red lines, that's the axons, and those are in channels. That's why you see it separately. You know, manage to culture individual neurons and image the individual axon. It's quite fascinating really to see this. Anyway, I'll turn off my video because my internet is not as stable. Okay, so moving on, next slide, please. So I would like to show you a few of their results. So here you can see the a part of the results where they showed successful differentiation of your functional motor neurons from your human IPs or iPSCs. So in this work, they did immunocytochemical and electrophysiological characterization of your human motor neurons differentiated from your human IPs. So here on the left top, so that's your image A, you can see the phase micrographs of your IPs derived motor neurons. Excuse me. And from B to D, those are neurons expressing your motor neurons marker. And then, so you have here islet1 and HB9, and I think SMI32 as well as the pan-neural markers. So these are just markers, neural markers, MAP2 or beta-3 tubulin. And here on the left or right sides, you have the ENF. Those are actually patch clump recordings from the differentiated neurons at day 27. So I've also tried to develop this patch clump methods, but it's quite complicated. So a little movement with patch clump recordings would give you a different recordings. So you have to make it really stable. So this one, they did it for at day 27. Yeah, they demonstrated repetitive firing during current clump, indicating, of course, excitability. And then they tried to use glutamate. So this is F. This is your image F. So that's application of your glutamate to the patch neurons, which, as you will know, your glutamate could elicit active firings, confirming their responsiveness to your glutamate. This study is quite impressive, not just because I have a bias when it comes to neuroscience, neurology, and neuropsychiatry, or anything that has something to do with the brain. But this particular study is really impressive because of how they establish a functional human-based ALS NMJ model. So we've discussed about disease modeling, so they did this by integrating ALS patient-derived IPs and motor neurons. So that's impressive for me. And primary human skeletal muscle into a compartmentalized chamber. And as I mentioned before, the NMJ function was analyzed for clinically relevant phenotypes. The MNs derived from ALS patients here demonstrated previously observed pathological phenotypes, including increased axonal varicosities, reduced axonal branching, and then your growth rate, and what else? Of course, increased excitability, how could I forget that, to validate our differentiation methodology. So when they introduced into the NMJ chamber system, they also form functional NMJs with healthy muscle. So all these things that they did were quite impressive. So they managed to model that. They managed to model the ALS NMJs and also show distinct functional deficits, including a reduced number of functional NMJs. Of course, you reduce fidelity and increase fatigue index. So if you're familiar with those results. And this, of course, corresponds, the most significant part is these findings correspond to clinical symptoms observed in ALS patients, such as muscle weakness, fatigue, and wasting. And then the BOC's results from evaluation of potential therapeutics in this in vitro model could be used to predict other clinical outcomes for either common interaction or screening potential drugs or interventions for ALS. So that's, to me, is quite impressive. So let's take a look at the next study on body on chips. So another interesting work on POCs is that of cancer. I think you are quite aware we have a lot of NIH funding just for cancer because it's been killing people. So cancer being one of the bottlenecks of mankind, and understanding them has been elusive, depending on the type of tumor. Metastasis essentially represents a highly organized and organ-specific process with multiple destinations, such as the lung and the liver, or wherever it reaches another organ. There are multiple steps of basically hematogenous metastasis, including, of course, the degradation of your extracellular matrix. So I'm not sure if you guys are familiar with cancer studies. And then you also have the intravasation into the blood vessels, surviving in the circulation, and then into the distant organs. And these individual cancer cells, which are involved in your metastasis, they detach from the original tumor, and then they then circulate through the bloodstreams. And this is what you call your circulating tumor cells, or your CTCs. And this is a pressing concern. It's considered responsible for most metastases. And that's why I chose this particular work, because they look into simulating circulating tumors inside the chip. So you can see here on the left side is essentially the schematics of your microfluidic device. So this is essentially the reconstitution of the microfluidic model. The design of the microfluidic model here is composed of 4 layers. Can you see the 4 layers in there? And so you have 1 layer of basically glass substrate, then 2 layers of your PDMS membrane, and 1 porous membrane. So the porous membrane is sandwiched between your 2 PDMS layers. These are 4 parallel branch microchannels mimicking vascular microvessels on the top PDMS layers. So these microchannels join together at the syringe pump connector at one end, and then the cell reservoir at the other end. So there are 4 organ chambers on the bottom PDMS layer. After the top and the bottom PDMS layers were sealed together, you will have here the inlet and outlet of each organ chamber, where you can punch through from the top to the bottom layer. So it's quite tricky. The tricky part here is the alignment. So I'm hoping that, someone will actually develop a really good and stable microscope with an alignment that actually would be able to bond the PDMS together. It's because sometimes you can bond both to PDMS without even treatment, without plasma treatment, for example. But in this case, if you have another membrane in between, it is quite hard to bond it without leaking, without treating it. So a good alignment system with probably a plasma inside it could do the trick. So we tried, actually, me and my colleague tried to manufacture our own microscope with a liner and try to bond several PDMS for, if you have 3D or multiple layer PDMS. It's quite tricky, especially if your resolution is quite high. So just imagine if you have, let's say, 2 micron or 1 micron or even in a nanometer size microchannel, and you're trying to align that. Anyway, moving on, let's look at some of the results for this study on organ-specific metastasis of circulating tumor cells. So here, in the next slide, yes, thank you. Here you have chemokine, yes, CXCL12. So some of you might know that chemokine, CXCL12, is involved in metastasis via binding to CXCR4. I'm not an oncologist, so it's not really my expertise, but this expressed on cell membranes in a wide variety of tumors. So in this particular study, they did flow cytometry and immunofluorescence staining with your anti-CXCR4 antibody indicated by your, here should be indicated by the one on the top, so the image MCF7, and then you have the MGA MB231 and ACCM cells expressed in CXCR. So this allowed them to determine the CXCL12 induced metastasis of these 3 using the microfluidic model. Okay, so if you look at the image, the ones below, so those are your detected flow cytometry and immunofluorescence staining. So the red should be your CXCR4, and then this is expressed on your cell membrane. And then you have the DAPI, your blue, which is your cell nuclei. Okay, and your image B, this one, you have the CXCL12 induced CTC metastasis in the microfluidic model. So you have the concentrations of CXCL12. I think they vary the concentration, 0, 25, and 50, and then 100. Yeah, I remember that because it's an increment of 25. So times 2 times 2. From the upper to your lower chambers. So you have here the MCF7 cells on the left side, and then your MGA MB231 cells in the middle, and then they're labeled again with cell tracker, your green, and then your ACCM cells are on your right, and it's labeled with a cell tracker, which is red. So another important aspect is, of course, if you can see this, you can image it, that's all good because you can see it, but then you should be able to quantify it. And this is exactly what they did at the bottom image. So your image C. So here you can see the quantification of adhesive cell numbers. So the number of MDMB231 cells increased significantly with the 50 and then 100. However, the adhesion of your ACCM cells significantly increased only with 100. So there is some sort of threshold. So what are they saying essentially? So they essentially found that some of the flowing tumor cells stopped and attached to your UVIC cells when they pass over the top organ chambers containing your CXCL12. So the number of attached tumor cells gradually increased with CXCL12 concentration, which is quite obvious. But of course, in science, you always have to prove it with the data. Furthermore, you can see that the arrested MGA MB231 cells significantly increased at 50. So like I mentioned before, there is like some sort of threshold. And yeah, so this is really cool as well. I'm not an oncologist, I'm not into cancer, but it's really good to see that you can actually emulate that inside the chip, and you can look at how the tumor cells get distributed to various organs. Now another application is, sorry, next slide please. Another application is your long-term co-culture of 4-organ system. So here, they did a 28-day culture, I believe. Give me a few seconds to just drink some water. Okay, so where were we? Yes, okay, so this is systemic absorption and metabolism of drugs in the small intestine. Metabolism by the liver as well as excretion by the kidney are key determinants of efficacy and safety for most therapeutic candidates. I think this is the essence of this chip. However, the systemic responses of applied substances lack in most in vitro assays. So what they did here is they developed a POC maintaining the functionality of your 4 organs over 28 days in co-culture. So in this particular illustration, you can see the microfluidic 4-organ chip device. I don't know if you could see it clearly, not in A, but probably in B. So there is a 3D view of the device here. So if you look at it closely, the device, the 3D view of the device comprised of, this is polycarbonate cover plates. And then you have the PDMS glass chip accommodating a surrogate blood flow circuit. So that's your pink. And then an excretory flow circuit, that's your yellow. So yes, and then you have here the numbers representing the 4 tissue culture components, compartments or components for your intestine. And then you have the liver. So 1 is the intestine. It looks a little bit like a villi. It's so cool. Even you can see that in this image. And then you have the skin, you have kidney and your tissue. So there is a central cross-section of each tissue culture compartment aligned along the interconnecting channel. Sorry. Then if you look at image B, that shows you the result of just their evaluation of fluid dynamics. So the top view of the 4-organ chip is also more layout here, easy to see, illustrating the positions of your 3 measuring spots as well, your A, B, and your, where's that? Yeah, your C, A, B, and C in the surrogate blood circuit. And then you have 2 spots for your excretory circuit. That should be your D and your E, okay? Then, of course, at the bottom right, you have the graph that show the average volumetric rate plotted against pumping frequency of the surrogate blood flow circuit and the excretory circuit. So it's good to do a lot of fluid dynamics troubleshooting for microfluidic devices, even with devices which are not as complicated as this one. Okay, so essentially here, what the authors did include integrating preformed human intestine and skin models into the 4-organ chip on standard cell culture inserts at the size. I think the size here is 100,000-fold smaller than their human counterparts. And then they incorporated a 3D-based spheroid equivalent to your 10 liver lobules, which mimics your liver function. And finally, they have this a barrier segregating the media flow through the organs from fluid excreted by the kidney, okay, and then to your, membrane covered by a monolayer of your human proximal tubule epithelial cells. It's quite cool, actually, and it's not obvious in the image, but in this study, they used a peristaltic on-chip micropump to ensure pulsatile media flow interconnecting the 4 tissue culture compartments through your microfluidic channels. And then they have a second microfluidic circuit that ensures drainage of your fluid excreted to the kidney epithelial cell. So basically, they also try to, in this particular, also try to assure that, the physiological fluid to tissue ratios are maintained. And if you look at the results, so these are just a few of the results. I like this image in particular. So next slide, please. They did an in-depth metabolic and gene analysis that revealed the establishment of your homeostasis among the co-cultures within, I think, 2 to 4 days, and then sustainable over at least 28 days. So here you can see so your A image A is for, so that's red, so that's CTK19 expression in your small intestinal epithelial tissue. Yeah, that's so cool, intestinal epithelial tissue. And then you have the green ones. So whenever it's fudgety and muddy, it reminds me really of liver, which is, in this case, is a liver aggregate. So that's here. And then you have an epidermal marker, CTK10 green on your C image. And then you have the red one, your CTK15. This is for your skin biopsies. And then you have here the image D, which is a 3D image. This is basically your transporter your NAC ATPase, if you're familiar with that, in proximal tubule epithelial monolayer. So what does this image really mean? It means that at the end of 20-day co-culture period, they managed to investigate the tissue architecture of the different organ equivalents using immunohistochemical staining. So the human small intestine barrier exhibited a perfect physiological polarization as shown by your CTK19 expression along your crypt villi axis. So you see that? It's really cool. And I mean, when I saw this, when I was reading the paper and saw the image, I don't even have to read the description to realize that this is really a villi. So, and then you also have here, as well the down regulation, which decreased the number of microvilli and deranged the targeting of the apically distributed protein. So they managed to show this in their results. So furthermore, this chip also showed the three-dimensional spheroids of your hepatocytes co-cultured with your human stellate cells, the fibroblasts of the liver, preserved their initial well-defined 3D architecture. That's the main purpose of your BOCs, mostly over the entire co-culture period. And then the skin biopsies reveal a fully functional one, proliferative and differentiated epidermis, as your staining would show. And then you have the culture at a physiologically air-liquid interface that facilitates the maintenance of your stratified stratum corneum. So that's providing your in vivo architecture for topical substance administration. So then you have the NAK ATPase at the basolateral surface of your human proximal tubule cell layer, which indicates distinct transport functions of your sodium-potassium pump during the 28-day co-culture in this particular device. So it's really cool to understand the techniques that they use to look into the device and to see the results that within 28 days you can manage to have 4 organs working. Okay, so moving on, we have another, I think we still have 2 more BOCs. So this one is, it reminds me of, actually it reminds me of a couple of episodes from the series House MD, so if you're familiar with that. Wherein there are, many mysterious cases, which has a common ending, really, systemic toxicity. So our current in vitro assays do not fully represent physiology, as I mentioned so many times now in this lecture, kulit ko lang din eh, often lacking synbiotic metabolism. So in this work, the authors use functional human multi-organ systems containing eyepiece-derived cardiomyocytes and then your primary hepatocytes maintained under flow using a low-volume pumpless system in serum-free medium. And so you see here that the functional readouts for contractile force and electrical conductivity enable the non-invasive study of your cardiac function. So the presence, it's so frustrating to not be able to use my pen, because at least I could draw and doodle and show you which side of that. So the presence of the hepatocytes in the system induce cardiotoxic effects from your cyclophosphamidine, and then reduce them for, I think they did several drugs here, terfenadine, due to drug metabolism, as expected from each compound's pharmacology. So it's a pharmacological study looking into the cardiotoxicity of this compound. So in this particular illustration, you see the co-culture of cardiac and cardiac and liver cells, and then you have your microfluidic system, which is a pumpless microfluidic system. So more importantly, let's take a look at in detail the device. So here you have a heart lever MFD, or microfluidic device, produced with two outer housing of thick transparent PMMA. So like I mentioned before in the previous lectures, you could use, there are several other materials that you could use for your microfluidic device. And one common one is PMMA, although we've used PDMS for many studies, but PMMA, you can also use that. And then you have here a thick gasket of, this is a silicon PDMS sheet material to define your flow path, and then the positions of your biomems chip. So you can see that the CMEA there with the amplifier, the right side image. So a schematic of this microfluidic platform and interface used to measure the functional activity essentially. And then you have here the two, this is a laser cut acrylic layers. Yes, I think this is, two laser cut acrylic layers, house 2-layer cut of your PDMS gaskets that define your microfluidic flow pathway, and then compartments for each chip. So you have that in A. And then you have here, your cells were cultured in respective surfaces. So you have the hepatocytes on your glass cover slip. So that's your compartment 1. And then your cardiomyocytes on cantilever, compartment 2. Okay, and then your MEA chips, compartment 3. Okay. And then you have the medium exchange performed through both your reservoir, so that's your R1 and then your R2. And then the drug is added through your R1. Okay. And then you have here an elastomeric electronic connector, which was incorporated into the housing to interface the embedded microelectrode array to a custom printed circuit board for electrical measurements. You have a stainless steel wire electrodes, that your AM systems, embedded in your PMMA housing. Here in this particular study, they use a customized microelectrode array chips, which they designed as 2 rows of 5 electrodes. Each, this is around, well, you can see the calibration. That's, I think the electrodes were like 80 microns diameter and then 1,000 microns separation. Okay, for measuring your electrical activities. Signals on the MMA chips were recorded using an amplifier via a printed circuit board. So you can see there, connector in your C. Yeah. And then of course you measure the cardiac contractile function using the layer deflection based apparatus. So that's your D. And the drug compound quantification was also performed. So they did an HPLC of that. So that's your E. Okay, and more results on this particular device, pan-on-a-chip device. So in this slide, in the next slide, yes. In the next slide, I'll show you some of the heart, liver system, and cardiac outcome upon cyclophosphamide treatment. So how cyclophosphamide, they use 9 millimolar concentration, significantly affected the cardiac function, so electrical and mechanical, in the presence of your hepatocytes. So this is shown in this figure. So you can look at it from A, B, C, and D. So that's the green. And then your cardiac conduction velocity, spontaneous beat frequency, and contractile force were strongly reduced within the first 24 hours after cyclophosphamide addition. So this you can see as well in this graphs A, B, and D. Then there is an increase in your MISI, so indicating that you have a QT interval prolongation. So without the liver compartment in the system, so basically what it's saying is that without the liver compartment system, the cyclophosphamide is still affected the contractile force at 9 millimolar or at this concentration with decrease. However, your cardiac function actually recovered after 72 hours. While if you have the presence of your hepatocytes, your contractile force remained compromised with decrease. Yeah. So here, these results essentially suggested a potential metabolic activation inducing your cardiotoxicity. So you know that your human eyepiece derived cardiomyocytes, electrical and mechanical activities are also shown here. They were evaluated non-invasively before and after 24 and 72 hours of an acute administration of your cyclophosphamide, with and without your hepatocytes. So it's quite cool. This platform recapitulates primary aspect of your in vivo crosstalk between your heart and liver and enables them to do pharmacological studies. So the liver, as you know, transforms synbiotic compounds to improve their elimination from the body. So consequently, this transformation may cause changes in drug bioavailability and function and even degradation. So in this sense, the effect of the liver may generate metabolites that are more toxic or more effective than the parent drug. So this is essentially what they did. So a system like this, a system that enables the co-culture of human cardiomyocytes with hepatocytes would be a key development in the human on-a-chip system to enable the study of induced cardiotoxicity upon hepatic metabolism of compounds as well as hepatic toxicity. So these types of BOCs enables non-invasive readouts of your cardiotoxicity of drugs and their metabolites. It is an integration of metabolic function in toxicology models that can improve adverse effects prediction in preclinical studies and many more, actually. Okay, so let's take a look at the last one. So the last one is another toxicity study. So I've mentioned before, again and again, drug discovery, drug screening. And one of the critical aspects of drug screening is toxicity tests. So is it cytotoxic? So the high failure rate of drugs at late stage of development contributes significantly to this increase in costs, as well as delays in the development of new and more effective clinical treatments. So in this work, the authors report a functional human model to evaluate your multi-organ toxicity in a 4-organ system under continuous flow conditions in a serum-free defined medium utilizing a platform for 14 days. So this one is the 14-day study. But still remember this. This is, I think, the last paper that I read last week. So here you can see a schematic overview of your system indicating the location of each cell compartment within the fluidic platform is shown in your figure 1A. It would have been so easy to just draw it. Then the specific tissues in these chambers, the system is modular and reconfigurable, and that organ modules can be substituted in the device. So, and then there is this gravity-induced flow. This was enabled by utilizing a rocker platform to generate shear stress on your cellular monolayers within accepted range. So there are the cells could also be stressed, mechanically stressed. And flow as well could stress the cells. You can determine the flow rate and shear stress on the cells by the choice of your tilt angle. So, and then here you can see also schematic view of the compartments at the bottom, and then the 2 holders for the separate culture devices. And they also did, as I mentioned, shear stress distribution, which is on the right side of the image. I'm losing voice. Okay. Okay, so further results. More results on this. Like I mentioned, what they did is a multi-organ toxicological work using compounds like, here they've used DOX, I think. So DOX and ATR, yes, DOX and ATR. So that's doxorubicin and then your atorvastatin. Quite familiar with DOX and atorvastatin. These are the drugs that my grandparents are taking. Anyway, so they've used 5 micromolars and then 100 micromolars. They also tested, I think, valproic acid as well, BPA. And then your acetaminophen, your APAP. So what they did is basically just a toxicity trial. So the toxicity trials were initiated after 5 days under flow in the pumpless system and lasted for 48 hours. Now you have the drugs were administered through the reservoir closest to your liver. And then the cultures were compared to untreated controls at 7 days in the flow system for both your function and viability. And then the controlled 7-day cultures, you have here the bright field microscopy images indicated the normal morphology of your different cell types in the microfluidic platform under flow. So that's your A to D, A, B, C, D, yes. That's your normal morphology. And then your examples of your cell survival and expected phenotype after 7 days in co-culture were also confirmed using immunocytochemical staining. So the immunochemical staining is your, of course, your E, F, G, and H. So in this work, they demonstrated a 4-organ system with continuous recirculation of a serum-free medium that mimics human response to 5 different drugs for at least 14 days. The system measures electrical and mechanical response to various drugs as well as metabolic response. The design uses a pumpless system, which is simple, mechanically reliable, and low cost. And also the system, like I mentioned, is modular, reconfigurable, and the organ systems tested, both the liver, your cardiac, neuronal, and muscle, represent the most important organs in terms of side effects leading to drug candidate failure. So these are very common organs that we do when we're dealing with cytotoxicity in the lab. The results obtained from this work indicated that this human 4-organ in vitro system is a viable tool to study. So that's one of the breakthroughs for this paper. And that you have the organ-to-organ communication. So they prove this. You have drug toxicity, and they have the noble drug compound effects in humans for predictive purposes. So if we can do this with several or hundreds or even thousands of drugs at the same time using just several of these chips, then that would save a lot of money. So this generation of novel tool improves the predictive power of preclinical efficacy and toxicity studies. And this is substantial benefit to us, to the health industry as well, and will aid in the generation of human-specific target compounds with a significantly higher safety profile. So this is what we're trying to achieve, and this is also one of the reasons why I came back to the Philippines to do the BSP, because we have many, many sources of potential drugs and that we haven't explored yet, but other countries have already started exploiting. So we have to take advantage of our diverse fauna and flora and start drug screening. And if we develop platforms like this, which are low cost, then we could achieve that hopefully in the next few years. And so with that, I end this 3 lecture series. Thank you for listening. I could talk fast sometimes when, you know, my mouth is too slow for my brain. So sometimes, sometimes I talk really too fast, especially if I've read the studies quite recently. So I hope you enjoyed it, and I hope to see some of you in the next couple of weeks, I believe, Sir July, for the training. A few of you actually, because there is a limited slots for the training. Thank you, everyone, and I hope you enjoy it. If you have more questions, you can email me. I'm a bit too organized for my own good, I believe. So Sir Jampong actually is showing the email addresses. Yes, my email addresses. So for specific research, you can contact me in those emails. And the moment I read it, I usually respond immediately. If it's something of interest to me, I respond right away. So yes, feel free to ask more questions after this Q&A as well.
Okay, do we have any questions? So far, no one has Anyone? You may type your questions in the Q&A.
This one is complicated, so probably I bored them to that. I must have bored the participants to that with all the images. But those are so cool, guys. If you've read those papers, you would be so amazed as to what they could do with microfluidic device, and something that I am hoping we could achieve here in the Philippines.
Yeah, that's exactly true, Jal. I mean, all of us, for example, in my case, when I was making my own device back in the US, it was just, the idea was just to, try to understand the dynamics of how probably a stone, like a kidney stone material, would grow in a confined space, right? Because that's normally the situation that you have in the body. Yeah, and you just have to follow. You can start with simple systems, and then you can slowly build up on that. You can slowly understand and visualize what's happening. So I think no one has a question. I think Sir July would like to make some announcements.
Yes. For those who are asking for certificates of attendance, especially for the 3 webinars, the certificates are still being finalized. So in case we will be sending the certificates via email, so we will be issuing digital certificates instead. So kindly wait as we issue those certificates. And as Dr. Fojas mentioned, so for the future activities of hers as a Balik Scientist, so she's preparing trainings. So however, we are limiting it to I think a maximum of 20 people. So invites will be issued out later on. So we're still finalizing the details. So I think there's a question. Okay, Sir Brian, I think there was a attendee who submitted a question.
Okay, yeah. All right. So the question is, are there cases in the human organs that take advantage of turbulence? So, yeah, which organ?
I see. Yeah. Well, of course, with the blood system, we want it to be as laminar as possible. But I think one organ that to me could take advantage of turbulence or eddy is your stomach, because the more that it circulates and mix your food, the faster the dissolution of the food. So that's one. But it reminds me of who else worked with Dr. Muriel, Sir Brian? No one in this panel. Not one in this panel.
Joey,
because
Miguel.
Sir Joey. Oh, they're not in this Zoom call, right? Because
yeah, they're not in this Zoom call.
The moment that I heard about the word turbulence, it reminded me of my master's work wherein I modeled the circulatory system and how turbulence happened if you have atherosclerosis, if you have the narrowing of your arteries, which happened to my grandpa, sadly. So there is turbulence there because there is eddy, and that causes problem. But yes, so taking advantage of turbulence, I believe the stomach could do that for us. Mixing, better mixing, faster mixing.
Okay. Other questions? Okay, so Sir July, you may want to continue with your announcement.
I think that's all for now.
Okay. All right.
Yeah. So
yeah. Okay.
In case you have other questions you want to raise to Dr. Jal, so maybe you can message the Department of Chemical Engineering Facebook page instead, so we can forward the questions to Dr. Jal. For those who will be requesting recordings of the 3 webinars, I don't know if we can request a copy through the University Computer Center, but in case Streaming is available via the Department of Chemical Engineering Facebook page. The first two webinars are available on our Facebook page. Okay, so if you want to again check the webinars on the first webinar about organ on a chip, or the second webinar about planting chip, and for today the systems on a chip. However, for this session or for this webinar, there apologies as there was a 15-minute delay on the live stream via Facebook. So hindi siya talaga nasimulan. Okay, so Dr. Fojas, maybe you would like to add something about the trainings. Did you already
yes.
mention the prerequisites to the training?
Oh, no, not yet. Yes. So
Yes.
there is a bit of a prerequisite. So I would recommend that graduate students apply for it. I'm not sure if the undergrad has the advanced maths to be able to take on computational fluid dynamic aspect of microfluidics. So we will be using finite element methods in there. But we will be using softwares like COMSOL Multiphysics, which apparently we don't have license to. Yes, so I would recommend for those undergrad who would like to take on the training that you do a bit of advanced reading when it comes to maths. So finite element methods would be good. But mostly, yeah, graduate students are more than welcome. Faculty also are welcome. By the way, so you mentioned that this is on Facebook, and is it in public? Could you chop the email portion, because that would mean that everyone, the entire public, would have access to my emails to prevent lang the spam. Kasi the email, my Harvard and Cambridge emails are almost full. So baka lang if people are sending emails na not related to microfluidics or anything. Pwede ba yun, sir? Pwede bang i-cut yung live na hindi mapakita yung emails to the public?
We'll check if it is possible to edit the videos, since it's already available to the public na eh. Okay, we'll just check na lang. Okay.
Sorry. Hindi ko na-realize. Yes, yeah.
Okay.
Hindi ko na-realize kasi the emails are all there. Anyway, yeah, feel free to send me an email for those of you who are interested. And yes, looking forward to the 20 students.
Okay. For the I think there will be a part of a simulation, part of the training, right?
The first training, yes. Yeah, the first training would have some simulation process. And
can you recommend specs for the laptops or the computers that they will be using, since they will be using software to run the simulation?
Oh, I see. So that is if they have the license for COMSOL. I would be doing a discussion and then giving lectures, and then probably I will just send them some sort of a problem set that they could try at home. But so far, specs, so it depends on the design that they would come up with. But if we're looking at simple things, then a MacBook Air could do. The MacBook Air 26 version could do, because that's what I'm using at the moment. MacBook Air 2016 works well with simple designs. So, but if you're using my complicated ones, which we shouldn't really focus on during the training, especially if they're only starting. Yeah.
Mm-hmm. Yeah. So there's a question. The training is online. Oh, wait, there's another question. Let me just check. Okay. Yeah. There's a question here. Will it be required to have medical background before joining?
Oh, not necessarily. It would be nice to have a medical background, but it's not necessary. The necessary one is the applied maths background. You know, with maths, you could understand anything. Kidding. I have bias, but yeah. Medicine, you could take it after maths, but taking maths after medicine is, yeah. So no, it's not required. Medical background is not required.
So this is open, Jal, to, for example, if the person has a medical background, right, to anyone,
yes,
right?
It's open Send to them, yes. If they have a medical background, it's okay, but they need to have the maths background. And most MDs in the Philippines don't have that maths. That's the problem. So if they're a medical doctor and they have the maths, that's good. But if they're just a medical doctor with a Bachelor of Science in biology or psychology, it would be difficult for them. So, no.
Okay. So what will be the option later on? I mean, would it be possible for them to, get involved with someone?
Yes.
Who probably, okay.
They can collaborate probably, yeah.
Okay, help with that one. Okay. All right. I think that should be it. If there are no more questions, the guidelines will be coming from later on. You will be sent out. I hope everybody was able to pick up a lot of things within this morning. And thank you again for coming. This has been, the 3-day session, the 3-lecture session gave us a picture of what's, what's out there. And the very nature is that these are all microfluidic. These are highly interdisciplinary studies. You cross boundaries, you go outside your comfort zone. So whether it's difficult, it's difficult or it's an easy, for example, crossing from medicine, going to the engineering, crossing from engineering, going to the medical part. Those things are, that's part of the process. And I think one thing that you should be able to pick up is that if you're ready to learn, kahit siguro mahirap siya, you will be willing to cross that boundary. And that has been the training that you should have had way back, and you still have now, that idea of lifelong learning. Oh, there is a pahabol na question. How do they sign up for the simulation tutorial sessions? Will the invitation be emailed to all lecture participants?
Maybe I can answer that.
Okay.
If you have expressed your intent to participate in the three webinars, so we rolled out a sign up sheet before. So maybe we can base them on the email addresses sent. So based on the sign up, the sign up sheet. Okay. So since the details are still being finalized, so we will just be sending invites through the email addresses sent.
Okay.
Thank you so much. And thank you, Sir Jhalique and Sir Brian.
Okay.
Yes, everything is multidisciplinary, so collaborate, guys.
Yeah. There's always someone who will compliment you. It's like in a relationship. Tama ba, Jhal? Okay.
It's complicated, multi-parametric, and unstable. So you have to optimize that one.
Okay. All right. So I hope everyone learned, and then we hope to see you in the next sessions for the tutorial session. And whatever you learn there, you can actually still share it with other people who want to join the field and who want to also start participating in a highly collaborative environment. Again, maraming salamat po sa lahat sa pagdalo po sa Balik Scientist presentation po ngayong umaga. At muli, magkita po tayo sa susunod upang mas mapag-usapan natin yung mga iba pang aspeto ng microfluidic technology. And good morning again to everyone, and enjoy the rest of the day.
Thank you.